We are going to construct a profile. So for that profile construction, you can see here there is a generate profile option. I hope you are able to see this. There is a generate profile option. You can click this generate profile option and you can see now it will ask multiple sequence alignment data. So this particular tool requires multiple sequence aligned status required for profile construction. Again, the profile is also similar to pattern, but the pattern is a sequence like output. Okay, but profile is not a sequence like output, it's like a matrix like output. It compares each bases or each protein sequences amino acids, say first amino acid of our protein of interest with the next amino acids of the other second protein and third protein, fourth protein, fifth protein. Like this, they will compare each amino acid with other sequences and they will derive a score. With the help of the score, they will construct a profile. Okay, so for that purpose, it requires a multiple aligned sequence are required for generating the profile like a matrix output. Okay, now we will see this. For this purpose, we will download already available cholesterol output. Say for example, cholesterol aligned. This is the cholesterol aligned file we already got with the tool cholesterol W from genome.jp. So we will download this cholesterol aligned file. Now the file is downloaded. Now you go for this generate profile and using this ProSite browse option, you will select this cholesterol aligned file, open, and now we'll change this HMMR and then we execute it. Now you will get the profile. Since for that ProSite format, we should give like MSF format. For this aligned format is suitable for HMMR. HMMR, you can see here, HMMR, hidden Markov model. No, for that hidden Markov model, you will give this aligned file. Now you will get, this is what the profile. This is what the profile you will get it. So it will compare, it will provide the score for each amino acids of our gene of interest with the other gene of interest. So positions, this is what the positions it will indicate. This is the sequence, this is the number of sequence they are using, and they will give different scores and they will develop a profile. With this profile also we can search, like pattern search, we can also do profile search. So you can save this profile by clicking this option. Now the profile is saved, and you can also search by pasting any sequence. With this profile you can paste a sequence here and you can search this profile. So you can search particular sequence with this profile to identify whether the sequence of interest will match with this profile. So again, this tool is used to identify whether a particular sequence is matched with a group of sequence. So for that purpose you can use this. Now you can paste any sequence of interest and you can identify whether it will match with our sequence of interest. So if you prepared a profile with protein sequences, then you will paste here protein sequence. Mostly profiles are prepared with protein sequence only because that will give a good profile. If you use nucleotide sequence, the multiple sequence alignment or pattern construction or profile constructions, so they are not become significant. Only for protein sequence, this pattern construction, profile construction, they are become significant. So we will use any sequence of interest and you can paste the sequence and you can compare it with profile. See, now I pasted a sequence and then I'm going to compare the sequence with this profile, whether this sequence matches with this profile. So I'm going to search. See, now This is what the sequence I have given, and then this sequence is matched with our profile, and then they identified that this sequence is matched with this profile from sequence this much number to this much number. If you want to identify the detail, you can check the detail. So the sequence I have submitted is matched with the sequence given through the profile. So like this, we can identify. Again, see here, this sequence is not matched. Why? Because it is different organisms. Okay, so the other regions are having similarity, whereas this part is different. That is why it is not matched. So like this, we can identify any sequence of interest by using this profile search. Okay, so like this, you can search profile, you can build a profile,